Start the recording.
Okay. Well, yeah, let's get started and then, yeah. I just want to take a couple minutes. My name is Britt Anderson. I'm the president of the Minnesota Lyme Association. We're so pleased to have IGeneX present to us tonight to discuss the clinical and laboratory diagnosis of Lyme. As many of you know, IGeneX has been the global leader in the research and development of tick tests that accurately detect Lyme disease and other tick-borne illnesses for the last 30 plus years. Tonight we are joined by Johan Gumaelius and Dr. Joseph Burrascano. Johan Gumaelius is responsible for all the IGeneX sales activities in the central US, including education of IGeneX tests to physicians and other medical providers, and he regularly attends regional and national conferences. I first met Johan on the street down in Winona in front of Dr. Vercota's office years ago. Yeah, so it's nice to see you again.
It was great when we first reconnected. We knew that. She's like, "Didn't I meet you outside Dr. Vercota's office?" And I'm like, "Yeah, one of the few times I went down there because I never had to, because she was so well versed in all this stuff, so I didn't really have to educate her very much." So, but yeah, that's a funny story.
Yeah. And also we're joined by Dr. Burrascano. He is a physician who was an early innovator in the field of tick-borne diseases. He is well known for his pioneering diagnostic and treatment guidelines for Lyme and tick-related illnesses that has been circulated around the world since 1989. He's a founding member of ILADS. He continues to educate practitioners and the public on the clinical diagnosis and treatment of people with Lyme and other tick-borne diseases. As a result of his countless contributions to the field, he has authored and edited articles, book chapters, textbooks, book reviews, and more. He's advised the CDC, the NIH, and testified before the Senate. He also serves on the editorial review board for numerous medical journals. Dr. Burrascano currently works full-time as a project analyst and manager for a private biotech company. And now just a little bit about the Minnesota Lyme Association. We will be recording this presentation and posting it on our website in the coming days or week. Our website is mnlyme.org. At the bottom of the homepage, you can sign up to get emails about talks such as this and about our monthly support group. The support group currently meets the second Tuesday of each month at 6:30 on Zoom. And when we have speakers, they are typically held on the third Tuesday of the month, also at 6:30. Yeah, as Dr. Vercota said, if you could all keep yourself muted, especially with the storm that's going on when you're not speaking. And yeah, I'll hand it over to IGeneX right now. And thank you so much for being here.
Thank you. I'll hand it over to Dr. Burrascano.
All right, so let me do the screen share. Am I allowed? I guess I can do it.
I think you should, right?
Can
All
you
right, here we go.
Oh, excellent.
Look at that. All right, start at the beginning and
Joseph Burrascano, brilliant doctor.
where do I do it?
With the bottom right. Bottom right.
Well, no, that's covered by the bar. Let me do this.
Oh, it's covered by a bar.
Let me do this. Bear with me. Technical things. You see that? Can treat Lyme disease, but I can't fix my computer. There we go.
And
That's
just one thing, Justine, if you could mute yourself before we start, that would be great. Thank you.
Okay. I think I'm set. Okay. So again, thank you everyone for joining us tonight. I want to give a little bit of an announcement before I get started. I'm going to be going through things in very great detail on purpose. And I don't want it to be like one of these things and make you like turn off to this. But I understand that in certain parts of the country, and I'm being politically correct right now, there are a lot of people who deny Lyme disease. They deny people's illness. They ignore positive tests, or they don't even do testing for some patients. So what my goal here tonight is to give you not only the information that you might want and need as a patient or a caregiver, but also the scientific background that explains why we do what we do and the validation of all these different tests so that you have ammunition. So someone doubts you or doubts your test results, to say don't do it, you now have this as a background. And that's why I think it's important if Johan can have these slides available to you, because then you can go over them, show them to doctors or others who might doubt what's going on, because he'll be presenting kind of background that gives you the idea of what you need to do. All right, so start off by talking about how ticks, I made this term up, that ticks are nature's dirty needle, because, they live in the dirt, they drink the blood of all these wild animals, and they pick up a lot of different organisms, not just Lyme disease. So, the major ones that we talk about are the Lyme and relapsing fever Borrelia, the Babesiosis, Bartonella, all the different rickettsias like Anaplasma, Ehrlichia, and others. There are viruses, there's Tularemia, there's maybe even nematodes, which is a kind of a worm, chlamydia, maybe even some yeasts in it too. So it's not just Lyme disease. What we're going to do tonight is focus on the major ones. Now, this is proof of what I've been saying. IGeneX did a study they released in one of the Lyme meetings a number of years ago. Over 10,000 samples were tabulated, and they found that 37% had the bees in it, 32% had Lyme, almost 28% had relapsing fever Borrelia. We're going to talk about that as a separate topic because it's related to Lyme. A lot of them had Bartonella, Anaplasma, Rickettsia, but more importantly, 40% had 2, 15% had 3, and some even had 5. So this whole idea of the tick being able to give you a lot of infections at the same time is a really big concern. So we started by talking about Lyme Borrelia. Now, Borrelia is the name of the spirochete, but there's 2 major classes. There's a Lyme disease Borrelia and tick-borne relapsing fever Borrelia, and that's really a big deal I need to teach you about tonight. Now, why is Lyme controversial? Well, obviously, because it's very hard to diagnose. In many cases, they're missed. First of all, the symptoms are not very specific. Only 13%, according to Texas survey, recalled a tick bite. The rash has been seen in less than 50% based on the vaccine study of over 13,000 people. So that's a pretty good statistic. And you can still hear that everyone has a rash. Well, that's not true. The FDA approves Lyme test kits, and these are the ones that are available to you in hospitals and in big commercial labs. But all these FDA-approved test kits have to meet a certain standard. And that standard is a very poor standard. I'll show you the data on that, meaning these tests are massively insensitive. As many as half the cases of Lyme are missed based on these lab tests that hospitals and big companies do. And because of that, you can never say Lyme doesn't exist just because of a negative test. I've seen so many times where we ruled out Lyme disease because the test was negative. You can never do that. And, if the diagnosis is delayed or missed, a lifelong illness can result. Now, it's important to know about the history of this because it kind of sets the stage for the problem that we have with testing nowadays. So in the 1970s, a lot of complaints to the CDC that finally sent down researchers. And early in the 1980s, Willie Bergdorfer, who was a tick researcher from the Institute of Health, he discovered a spirochete in the ticks that he collected from an island off of New York State, off by the Atlantic coast, and it happens to be about 20 miles from Connecticut along the bird flying route, and we know birds can transmit or carry ticks. So they named the spirochete after Willie Bergdorfer. That's why it's called Borrelia burgdorferi. So a few years later, they finally developed some blood tests, and these blood tests all were based on this one strain of Lyme germ of Borrelia that he found in the tick from New York State. And this is now the basis of all the FDA-approved Lyme tests, the Lyme serologies that, as I mentioned, you get in the hospitals and in the big commercial labs. But nevertheless, I was there at the time. Willie and I worked together. We shared patients and so forth. We found these patients really looked like and had the symptoms of Lyme, but they had a negative blood test. They were seronegative. So my thinking, and this is relevant for now, is that a single strain of a single species of Lyme Borrelia from a coastal island in New York doesn't necessarily reflect what's going on in the rest of the country and around the world. But also importantly to note that this spirochete that he discovered, that they based all these blood tests on, came from a tick, not even a human being. So that right there sets the stage for why the basic tests are not so good. Now, just to make it even more complicated, the thing with Lyme disease is this germ Borrelia burgdorferi. Well, the one that was found by Willie Bergdorfer in New York, they call that Borrelia burgdorferi senso stricto. That's BBSS. You see over here in the top left in red. And they named that strain, that specific strain, B31, named of the 3 different doctors who discovered it, and it's a whole long story. But anyway, this is that one specific germ that came from a tick from an island off of New York, and that's called Borrelia burgdorferi senso stricto. But we now know that there are a number of Borrelia, not just Burgdorferi, but Californiensis, Mayonnaise from your neck of the woods, Sapsilia and Greenia in Europe and America, others. They also can cause Lyme disease. They're also carried by these ticks, and they're called this complete group, Borrelia burgdorferi sensu-lato, BBSL. Okay, so that's terminology that you may or may not have heard about before, but it's important to remember because it has relevance to the rest of the topic tonight. Now, the tick-borne relapsing fever Borrelia, it's called TBRF. The two that you might have heard about is one called Borrelia hermsii, another one called Borrelia miyamotoi. But these other ones are also present in ticks and in people in North America. So the key is that you need to be able to test people for all of these germs because they're all here in America. They're all here in our patients. But again, these in red, these two relapsing fever ones and this one red one, Lyme Borrelia, are the only ones that you can get a commercial test for. So if you have this one, for example, or this one, for example, there is no test for that in these big commercial labs in the big hospitals. But they are included in the testing that IGeneX offers, and that's the big difference, okay? Now, is it just some kind of vague lab thing? No, these are actual cases. What was done, 36 patients from the left column, is from a single practice in North America. These are patients who had a negative blood test by the big box labs, but they found that these 36 patients were positive on the IGeneX Lyme immunoblot, and this is a test we'll talk about because it does test all these. And they found what? They found Mayonnaise, Spilmania, Californiensis, all these different ones. They found one of the strain B31 that their test was supposed to pick up and didn't, this big box lab. But all these other they found, including 5 other cases they knew were there, they couldn't even identify what they were. Then there's another series from a different doctor's practice. 48 patients were negative on the Lyme test, but by testing for relapsing fever by the IGeneX test, they found all these other ones, including 26 cases that were there, positive tests, they couldn't even identify what they were. And when that happens, when they can't tell you the species, it often means that the person's infected with more than one. There's so much reactivity in the body that you can't separate them out. So here again, 48 cases of a negative Lyme test, but they didn't have the regular Lyme, they had relapsing fever. Here was a regular 36 where they only one had the regular Lyme, missed on every other test, but they found it on the IGeneX immunoblot. Now, relapsing fever, you need to know about because it's very common and it mimics Lyme disease. Here's a series, 543 cases of patients from the United States, not from Africa somewhere, but from the United States, who had all the signs and symptoms of Lyme, where the doctor thought they had Lyme disease and did Lyme tests. Well, when these were tested, a third of them, 32%, had antibodies to Lyme in their blood test. 22% did not have Lyme, they had this relapsing fever, and 7% had both. And if you look at the bottom in the map, where the Lyme is in the green and relapsing fever in the orange, they pretty much overlap. So not only did they overlap in the symptoms, but they overlap in where these diseases are found in our country. Now, here's another report. 90 patients who met the published in journals clinical case definition of chronic Lyme disease were not suspected to have relapsing fever. Of these 90 patients, 46 did have Lyme, 56 did not have Lyme, more than half. They had this relapsing fever infection, and 8 of them had both. Okay? So we're talking about the clinical things that define Lyme, and this goes for both Lyme Borrelia and the relapsing fever that I've talked about. 3 important things: multi-system, migratory, and cyclic. Multi-system means it involves a number of different parts of your body, the joints, the muscles, the nerves, the brain, basic symptoms like fatigue and headache. So in other words, if you have just a sore knee and nothing else is wrong with you, I kind of would doubt that's Lyme disease. But if you have several different things happening, that's multi-system. That's a clue. Another thing, which is even more important, it's migratory. Lyme is the only illness where arthritis and the nerve damage can come and go and move around. It's typical someone will have a really sore knee for a number of weeks or months, and it gradually subsides even without treatment, but then the wrist starts to hurt. Then they start to get tingles all over the place, but then it goes back to the knee again. No other illness does that. Now, this is also why Lyme patients are labeled as being a little bit off. They don't get rid of it because whatever the doc say my knee hurts, and they come back to me and say, "Oh, my knee is fine. Now my nerves are tingling." Then that goes away, and then it's my wrist. They start to think the patient's making it up, but no, that's a classic sign of Lyme. That's the migration. And the final thing, which is important also for the doctors to notice, is that Lyme germs don't grow in a steady fashion. They grow and then they go through a quiet period, and they grow again and go through a quiet period. And every time they recycle, they undergo a genetic shift. And that's why maybe every month the symptoms change. In classic Lyme disease, this is a 4-week cycle. So maybe 1 month you have a really bad week, and then you gradually start feeling a little bit better by the 3rd week, and by the 4th week you're sick again. In relapsing fever, the cycles may be shorter, maybe 3 weeks, in some cases 2 weeks. And that's why it's very important for my patients to keep a symptom diary and a calendar, because you can track that it's multi-system, it's migrating, and it's cycling. Okay.
Dr. Burrascano, can I interrupt for just a second?
Sure.
We're hearing some input that you're a little distorted or muffled sounding. I don't know if there's something with your microphone that could be adjusted.
I have earphones. Aha, let me try that.
Thank you.
Okay, is that any better? How does this sound? Is this better?
Yes. better.
Ah, now you're louder too. I'm going to calm you down. Okay. All right. Should I go back to the last slide or continue?
I think that's fine. Thank you.
Okay. All right. Thank you too. All right. So, the immune system. When you have a Lyme disease infection, it's caused by the Borrelia, and the Borrelia can invade, inhibit, and even kill the immune cells of your immune system, the B cells and the T cells. And it's known that Lyme patients have suppressed immunity. And this is the reason why. The Lyme is actually killing their immune system. And because serologic blood tests like the ELISAs, the IFAs, the Western blots reflect the activity of the B cells, if you have immune deficiency from Lyme disease, you can get a false negative blood test. Same thing true with the T cell test that you talked about before. So the result of this is a negative test, seronegativity on both the B cell tests, the ELISAs and so forth, and the T cell test. It also means the disease can be more severe and the treatment can be more difficult because your immune system's not there to support it. So the most important thing here is to use the most modern, most proven, and most accurate tests, because otherwise you're going to miss it. So let me talk about Lyme specifically first, and then relapsing fever. This is data from a study that was published in 2007 on the ELISA test. They found the overall sensitivity of Lyme, based on summarizing these 9 different studies, was only 49%. 49%. If you look at the columns here, sensitivity, the sensitivity was anywhere from 60, 50, 50, 29%. Here's one at 75. Now, why is that better? When you do testing with the standard ELISAs, there's a trade-off between sensitivity and specificity. So if you want to make the test more sensitive by turning it up, well, what happens is you start to pick up other germs that aren't Lyme. So it's less specific. You start to get false positives. So there's a trade-off between false negatives and false positives. So if you engineer the test to be pretty specific for Lyme disease, you're going to miss half the cases or more. Okay. Now, what about the more modern tests? And you hear about different labs have these new methods using microarrays and multi-wells. So basically, that's an adaptation of the ELISA that's just put in a different type of a machine. So the accuracy there has not been proven to be any better than this. Okay, that was done in 2009. Here's a study from Johns Hopkins published just a few years ago. They used the 2-tier testing, which is what CDC has recommended, and they used acute and convalescent paired samples. Now, that's supposed to be the highest standard for getting a positive Lyme test. We take someone who initially has Lyme, you do a blood test, you wait 4 weeks, and you do it a second time, and you want to see how it changes as the infection takes hold. Guess what? 53% sensitivity, pretty much in agreement with that study from a number of years ago. So even though this is supposed to be a very high standard test and was done at a great hospital, Johns Hopkins, again, the test was only 50-50. So now we talk about, how about a Western blot? A Western blot is supposed to be a more accurate way of testing for Lyme than the ELISA. Well, there are a lot of problems with the Western blot. You know, when it first came out, it definitely was a better choice than the ELISA, but nowadays we've moved on. Well, let's talk about a Western blot. Again, I don't want to get too technical here, but in fact, I do, because I want you to have this in your back pocket to defend what you do when you talk to your doctors, when you have to defend your position. A Western blot is made by taking a Lyme germ, put it in test tube, and let it grow in a culture, And then after they have enough, what they do is they break it apart with sonogram, they sonicate it. They break into many, many little pieces. And then they separate them out on paper. You see the picture here, using electrical charge. And what happens is if you put the drop of this solution of mixed up lime fragments on the paper and you put electrical charge, these different particles will start to migrate along the piece of paper. And the heavier they are, the closer they stay to the beginning, and the lighter ones move further away. So basically what this does is it separates out the different components of the lime germ based on their weight, their size, and molecular weight, okay? The problem is when you take a germ and you break it into many, many little pieces, you release parts of the germ that are specific for the lime, but you also release pieces of the germ that are not specific, that all germs have. Okay, so when you see these things migrate along on a Western blot, a lot of these are from a lime germ, but some of these could be from other germs, could be from viruses, could be from autoimmunity. Okay, so it's not very specific. And the other thing is, when you grow this germ in the culture, you're growing one species. Remember that slide in the beginning? I showed you all these different things can cause Lyme. Well, the Western blot is just one. So you're missing them right there. So how you identify these different fragments of the germ is where they end up on this strip of paper after electrical charge, after electrophoresis. And where they end up, they give it a number. And these are what we call the bands on the Western blot. There's band 22 and 31 and 34 and 39, and they migrate along. Now, this dark strip here, number 8, that's the test strip. This is the whole intact germ, and the ones here are the different patient samples. So let's say you have a patient sample here. This dark one, you say, well, that's definitely a positive one, and so is that one. What about the ones that are weaker? You know, what does it take to be called a positive on this? Okay, so when you do these Western blots, this separation out by the electrical charge is not very exact. You have a lot of non-specific things that migrate out that can give you false readings. How intense the darkness is how you determine if it's positive or negative, but the intensity often is related to this original culture, how good the culture was, how well it grew, the conditions of the paper test strip, the temperature, all these different things can affect it. So basically, it's not that precise. And the sensitivity has been shown to be about 50 to 70% for a Western blot. So it's slightly better than an ELISA, but based on all these different reasons, you can see why it's a very imprecise thing. So the CDC tried to make criteria for the Western blot to try and make it a little bit more accurate. But it's very important to know that they did not do this to help patients. This was not meant for clinical diagnosis. This was meant for epidemiologic surveillance to count cases, okay? So the CDC criteria, again, are for surveillance, not for clinical diagnosis. And for surveillance, they emphasize the test being very specific, so they don't miss any cases. I'm sorry, so they don't get any false positives, even at the cost of sensitivity. In other words, they want a true positive to be really, really positive. And yeah, we're going to miss some of them, but that's not important because we're only counting cases. It's nothing to do with patient care. So what their criteria do is they tell you which of these bands, these black dots, to consider to be important or not important, how many you have to have. But the problem is they included these bands, these numbers that are not specific to Lyme, so you can get false positives. They exclude bands that are specific, give you false negatives, and so it's just not accurate. But the point is, these not meant for clinical diagnosis, they're meant for surveillance. So if you want to be counted, you could use it. If you want to get treated, you don't. All right, so then comes what's called a two-tier test. So in the ideal world, you want a test that's very sensitive, but at the same time very specific. And as I showed you before, this ELISA test is this trade-off. So to get around that, they decide to make what's called a two-tier test. So you start with the first tier. The first test, it's 2 tests are done at the same time. The first one is supposed to be very, very, very sensitive, so you don't miss any cases, even at the risk of maybe getting some false positives. So if you get a positive, then you do a second test, okay? And the second test is supposed to be one that is as sensitive, but much more specific. And to make the statistics work, the first test has to be 99% sensitive. Second test has to be as sensitive, but 95% specific, okay? Those are the numbers. When it comes to Lyme disease, the CDC uses the ELISA as tier number 1. It's not 99%. I showed you it's like 50%, 53%. So you're going to already miss up to half the cases. Now, the second tier is either a Western blot or a different ELISA. And that's no better than the first. So you're also going to miss a lot of cases. And once again, this two-tier scheme from the CDC is meant for surveillance, not for clinical diagnosis. So if someone says to you, you don't have Lyme because you don't meet the criteria for a CDC two-tier, that's absolutely false. They don't know what they're talking about. This is a test for surveillance and not for diagnosis. And I know I was on the committee that came up with this thing 100 years ago. So here comes IGeneX, and they threw all this out the window, and they come up with what's called the immunoblot, and it's completely different. What they did was, instead of taking germs and growing them in test tubes and breaking them apart and smearing them out on pieces of paper, they developed laboratory-created antigens. Those little black dots you see on the paper, I'll show you another picture, were not made from germ smears. They're made from actual chemical processes where they look at the DNA components of the germ that they want to test for, and they make the antigens directly from that. They also have the ability to not only get very specific antigens that only come from the germ you're looking for, but they also can get them from a number of different strains and species. So you're not just doing that B31 tick germ, you're doing all the ones that are important. So once they've developed these antigens, what they do is they use a kind of an inkjet printer, kind of a printing process to print these onto the test kits so that you know exactly what you're seeing. So those little bands that show up, you know exactly what they mean because you printed it there. The location is not dependent on migrating with electrical charges. You printed it in a specific spot. The intensity of how dark or not dark it is not dependent on some culture or some chemical reaction in the testing process because it's printed. They know exactly how much they put on there. Also because these antigens are specific that they created, they're not from germs. You're not going to get a false reading because some other germ happens to be there in the mix that reacts with it. And the bands are more clearly displayed. Now, a lot of mumbo jumbo, but this is the actual thing. You've seen this before. This is the Western blot, the test kit, or the testing strip, the control. This is how absolutely clear that is. There's no comparison, no comparison at all. The positive control, you can see the negative control is clear. So there's no comparison. That's why the immunoblot is so much better because it's made from more accurate chemicals. It's made from specific chemicals. It's printed, not smeared. And it includes many different species. Basically, they include all the important species found in North American, Central American, and European species. Now, why is that important? Well, people travel, birds fly, and it's possible to get so-called European strains of Lyme in America. That's been proven again and again. In Lyme, you can get all the clinically relevant ones. In relapsing fever, the same thing. Notice that there are separate immunoblots for Lyme and tick-borne relapsing fever, and those two don't cross-react. So you can do an immunoblot for both. If one shows positive, the other one shows negative, then you know you have exactly what the test shows you because they don't cross-react. Also, it means if you're sick with Lyme and do a Lyme test and it doesn't show anything, or maybe it's not Lyme, maybe it's relapsing fever, so you should do the TBRF immunoblot. Now, I want to talk to you about validation. Everyone says how good their tests are. Well, let them prove it. This is some really interesting data that I wanted to share. This is on the table. They talk about samples. These came from the Center for Disease Control, from the CDC. Patients with early Lyme, like when they just show up with sickness, convalescent Lyme a month or so later, patients with late Lyme, neurologic and Lyme arthritis. It's been said that in Lyme disease, if you get Lyme early, don't even waste your time doing a blood test because it takes time for the test to show. And here at the CDC, standard two-tier test, it only picked up 20% of the cases for early Lyme. With the Lyme immunoblot, it picked up 93% of the cases. Never before in the history of Lyme disease has there ever been a test that you can use in early Lyme disease. How many times people come in from outdoors and a number of days or weeks later they start to feel sick? They didn't get a rash. They don't remember a tick bite. Is it a summer flu? I don't know. Or there's a rash, but is it a spider bite? I don't know. Is it ringworm? Now we finally have a test that has a 93% chance of picking it up based on CDC's own samples. When it comes to the established cases, it picked up all of them. Okay, so that's really pretty good. Now, the other thing is to say, okay, it's picking up all these tests. What about, is it specific? Are there any false readings? Well, they found that the sensitivity, as I said, is very high, but the specificity is at least 99%. Now, that's important for another reason. when you do testing for Lyme with serologies, you can do what's called an IgM, which shows up early, and then IgG, which shows up later. And the dogma has always been that IgM, which is the one that shows up first, sticks around for a few weeks, maybe a month or two, and then gradually goes away, even if the infection is still there, because it's an early response. And then the IgG appears and takes over. It's also been found in Lyme patients that a lot of times they continue to have an IgM even later on. Now, if you do IgM testing with a Western blot or an ELISA, because they're not very specific, some of these positive IgMs came from viruses or other infections, and the reputation and the dogma has been, if a Lyme patient has a positive IgM test later on in the disease, you can ignore it, it's a false positive. Well, that may or may not be true for an ELISA, but it's not true for the immunoblot. Here, again, it's 99% or more specific. So if you have a positive IgM, and a lot of Lyme patients only have an IgM, because Lyme, again, weakens the immune system, it doesn't allow this change from M to G. 99% of the time, a positive IgM means Lyme disease. Okay, so do not ignore a positive IgM. This is the thing you may have to educate your doctors about, because either they don't know this or they choose to ignore it. Now, how about the validation of it? This is a second test done, again, using samples supplied from the CDC, using interpretation criteria that IGeneX came up with, not the CDC two-tier, 10 band, 10, 5 out of 10 blots on the band, whatever. This is IGeneX's own criteria, which people criticize as not being that accurate. Well, here they are, CDC supplied samples. They had a 100% specificity, no false positives, despite using IGeneX's criteria. So those who say, don't believe IGeneX, everybody gets a positive test from IGeneX. No, they're 100% specific, not a single false positive in this series from the CDC. And in their own studies, at least 98, 99%. This test included, from the CDC, they found 90% of them were picked up, and that included all stages, early and late, males and females, young and old. Okay, so it pretty much confirms what was found earlier. Now, the problem is this. As good as these new tests are, there still are times when you can't get a positive serology or antibody test. Now, why is that? Well, sometimes your body doesn't have enough to be detected. Turns out these tests pick up antibodies. Antibodies are made by your body's immune system, by the B cells, and they act like magnets. They attach themselves to fragments of the germ to clear them out of the body. If you have so many germs, all these antibodies get attached, and the test can't pick them up when they're attached. So in an overwhelming infection, you can have a negative serology. And a lot of you might have known that the sickest Lyme patients are the ones that are least likely to have a positive test, and this is one of the reasons. Another reason, as I mentioned before, Lyme weakens the immune system. So if your body is not making enough antibody because of the bad B cells, then you're not going to pick it up. The final thing is, sometimes the germs hide from the immune system. A Lyme germ can hide like deep in your tissues, inside your cells, hiding into biofilms, and the antibodies may not even be recognizing it. So these are reasons why you can have a negative test. It's much less so with the immunoblots I've shown you with the statistics, but still there are cases where people with late Lyme, with weakened immune system, or they're on chemotherapy or steroids, they end up with a negative test, false negative, even with an immunoblot. So because of that, people are trying to do advanced testing using what are called direct tests. We directly look for the germ, not antibodies. And the two tests that are done for Lyme are PCR and culture. So what about a PCR? PCRs have been around for a long, long time. I think they were introduced in Lyme disease probably around 1990, maybe 91. What they do is they look for fragments of the organism's DNA. They're called nucleic acids. The problem is, we know that the PCR for Lyme is notoriously insensitive. The pickup rate is really low. If it's 20%, in my experience, that's a lot. And why is that? Well, in the bloodstream, there are chemicals, normal chemicals like heparin, your own DNA, and even the hemoglobin from the blood cells that inhibit the process that the laboratory uses to do this PCR. So the inhibitors can give you false readings that you don't show it. Second thing is that sometimes there's not enough germs or DNA in the bloodstream to detect it, especially because of these inhibitors. And the other thing is, the germs aren't always in the bloodstream. They come and go, especially in Lyme disease. They can be there intermittently. It's different times of day, different days of the week. So, you can take more than one blood tube. You can collect several blood tubes. You can do blood tests several times over a week, like 2 or 3 samples. That can help to increase the yield. You can use like urine and spinal fluid because they have fewer inhibitors, and you can do biopsies to try and get around this whole problem. But still, the sensitivity of PCR is really, really, really terrible. So what IGeneX has come up with to get around this is culturing. Now, culturing has always been the gold standard of infectious disease. If you have a urinary tract infection, you got to culture the urine. Basically, put the urine in an incubator and you see what grows. So now it is possible to do this for Lyme disease. They call it a culture-enhanced PCR. So they take the blood, they incubate it for usually a couple of weeks, and after 2 weeks, they analyze it. And if they find germs there, then they'll do a special PCR that they develop to look for it. By doing the culture, not only do you multiply the number of germs, but you also get rid of the inhibitors. So now the PCR can work. So the other thing that's good about this is you can get it for Lyme, for relapsing fever, for Bartonella, for Babesia, Ehrlichia, Anaplasma, and even the Rickettsias. And when these things are done, they look for 2 different types of DNA in their PCR. They look for the one that looks at the genus as well as the species. And that allows you to look for a whole variety of germs that you otherwise would never have found, because they're looking at the group of germs, the genus level, not just an individual germ. Now, how about the performance of this? During their development, all the positive samples were sent to an outside lab for confirmation. What they do is they take the fragment of DNA and they analyze it piece by piece, layer by layer, to find out what exactly is in it. And this is called sequencing, and that's the only way to be 100% sure. So when they developed this culture, every single positive sample was sent to an outside lab for sequencing. Second thing is, it's possible that the culture just had some DNA in there and maybe it wasn't really a live germ after all. So what they did was they developed a reverse Western blot, believe it or not, and they do that on all these samples because this would only show positive if the whole germ was there, not just a piece of DNA. So what came up? 100% of the time, the sequencing matched the initial PCR, and 100% of the time, the reverse Western blots confirmed that there was, in fact, an infection. So the specificity here was 100%. Sensitivity, though, is a problem. We don't know because, there's no gold standard. This culture itself is a gold standard, so there's no real way to compare it to. But if you compare the culture to a standard PCR, and I've seen this data, the sensitivity at least 10 times more sensitive, and maybe much, much more so than that. Now, just as an example, this is actual test results. I blanked out the patient's name who has immunosuppression, positive Lyme culture, but negative immunoblot. So this is a real thing, and that's why adding a culture to a serology like an immunoblot can increase the yield and likelihood of getting a positive test. Okay, I'm going to talk now about tick-borne relapsing fever because it can be confused with Lyme disease and has major implications for testing. And what I'm going to tell you tonight, a lot of doctors who are not Lyme-related will absolutely not know about, and you need to, at this point, educate them. Now, if you look at a textbook for relapsing fever, what is it? Well, they say you get an acute illness or really high fever, can be 104, even 106, severely sick. It lasts like that for a few days, ends with what's called a crisis, a terrible sweat, and you feel weak, you can even pass out. And then for the next several days, you start to feel better. Then after about 5 to 7 days, the whole thing repeats with another acute illness, high fever, several days, a crisis that ends it, and then it goes away. And these cycles continue a number of times, and eventually over time, they're supposed to go away and stop, and then you're over it. That's a classic description of tick-borne relapsing fever. Any textbook will show you that. However, as I showed you before, in a large number of patients, relapsing fever will present exactly like Lyme disease. And these patients will get tested for Lyme and not for relapsing fever. And the problem is Lyme tests do not pick up relapsing fever. So a lot of the seronegativity out there for Lyme may in fact be because you're testing for the wrong germ. As I showed you before on the map, it's been found pretty much everywhere. This map is not just from some government official. These are actual counties where positive tests were found by IGeneX. Look, even above the Arctic Circle, northern Alaska, they found this relapsing fever. So it's thought to be rare in the United States. Well, that's not true. It's found all over the United States, found all around the world. It was said to have a distinctive clinical presentation as cycles of relapsing. Well, that's not true. As I said, it can be exactly like Lyme disease. Another thing is, relapsing fever is supposed to only come from a completely different kind of a tick. They call them soft ticks. It's not an Ixodes tick. It's called Ornithodoros. Well, that's also not true. There are many, many species of hard ticks, like the Ixodes ticks, that harbor the relapsing fever. And just to make it even more of a point, these soft ticks are present in many parts of the United States. And before last few years, people didn't believe that, but in fact, it's been found to be true. So let me talk to you about these soft ticks because they're really wild. First of all, they're called ornithodoros. These ticks, unlike the ticks that give you Lyme, the ticks remains attached only for 5 minutes to 30 minutes, and then they drop off. So it's not like, oh, the tick is still on, you come in from outside, a few hours later, and you find this tick on you. No, it could have already bitten you and dropped off. Another thing, by studying mice, where they give them infections, this tick bite can transmit infection within 15 seconds. So long that you have to have a tick attached 5 hours, 6 hours, 12 hours, whatever that people are telling you. For this type of infection, 15 seconds. And I can't tell you how many times I've had patients say to me, I know the tick wasn't on me more than a few hours because I checked myself and there was no tick and I had the bite and had the rash and I got sick. Or maybe it wasn't Lyme, maybe it was this. Once these ticks get infected, they remain infected for the rest of their lives. Their progeny are born infected because the eggs themselves carry the infection from one generation to the next. So it means they are their own reservoir. They don't have to like feed on a mouse or a deer or some other animal to acquire the infection. They have it from birth. Now, the last thing is, it's been said, okay, this is a soft tick. It's a very strange, unusual looking tick, and you can easily tell it apart from a Lyme disease tick. Well, I'm going to show it's not always true. On the left here, you see what's called the ornithodoros tick. And if you look at this, yeah, it's kind of pudgy and fat, little fat potato looking thing. And I don't think you'd confuse this with the Lyme tick. However, this is an Ixodes tick, the tick that transmits Lyme disease by bees and so forth. Once it gets engorged, they look pretty close. I mean, look at this picture here of the Ixodes tick and this one of the ornithodoros. Look at this one, all engorged, a Lyme tick, an Ixodes tick, and here's the ornithodoros. So, if you're not a tick expert and you see something like this or like this, you're not always going to know exactly what kind of a tick it is. Now, relapsing fever. Okay, so it's in this strange kind of a tick. It's in certain parts of the country, certain parts of the world. Well, here's a study that came out from The Lancet, a very respected medical journal. They found relapsing fever has been found not just in soft ticks, but hard ticks also. They say, okay, well, we only get bitten by certain kind of ticks. We get a dog tick, a deer tick, an Ixodes, and whatever, the Lone Star tick. Well, they found that humans can be bitten by as many as 43 different types of tick species. Now, I highlighted a few things, just not to get too technical, but they found 6 relapsing fever species in 32 hard ticks and 31 soft ticks. The hard ticks are the Lyme ticks, the soft ticks are this and the ornithodoros type. So basically, it's 50-50. They found them in both. 4 species were found in both hard and soft ticks. Okay. They found what they call unclassified relapsing fever Borrelia. Germs, they don't even know what they are, but they know they're relapsing fever. As I said, they found human bites associated with 43 different tick species. You read in the literature certain types of tick germs, like relapsing fever germs or Lyme germs. Well, they're only in rabbit ticks, and rabbit ticks don't bite people. Or they're only in, ticks associated with cattle. Well, here's proof. From the Lancet, 43 different tick species can bite humans. And I have some test results to show you that actually where this was found. Now, they found that the soft tick was infected with 22 different species of relapsing fever. They found, however, 5 species in the Lone Star tick, 4 species in Lone Star tick, 3 in Ixodes, which is the Lyme tick, and 3 in Dermacenter, which is a dog tick. How many people have told me, oh, I had a dog tick on me, I don't have to worry about it because it doesn't contain Lyme? Well, at least 3 species of relapsing fever were found, so you're not off the hook. Miyamotoi is a fairly well-known relapsing fever germ that was discovered a number of years ago. They found it in 23 different types of hard ticks. They found it in 11 different Lone Star ticks, including 11 hard and 1 soft. They found this other one, Thalieri, in 7 hard and 1 soft. I found this Turricate, which is a turtle Borrelia, believe it or not. They found in 2 hard ticks, 5 soft ticks, and it's been found in humans. And to make it even worse, they found co-infection. You can have relapsing fever and Lyme disease in all different ticks, including 5 different species. They found both of them. Okay, so if anyone says that relapsing fever is not common, it can be easily distinguished from Lyme disease, you don't have to worry about it, they're wrong. One thing they say about relapsing fever, they discovered this originally in Africa. and sub-Saharan Africa, where it's very desolate and, it's poor living conditions. So the quote is, relapsing fever is considered typically a disease of outdoor enthusiasts and impoverished persons in primitive conditions. Well, the study found tachycardia in metropolitan areas. They found this ornithodoros tick in San Antonio, Dallas, Austin, which is the 7th, 9th, and 11th largest cities in the United States. University of Tennessee, 58% of their turkeys had relapsing fever in it. And they found relapsing fever species in humans, including Miyamoto, Hermsii, Lone Star, Parkeri, Trocardia, the turtle one, and many others. So again, it's a real problem. And if you don't test for it, you're not going to find it, and you're going to be missing a case. Again, I'll go back to the study I talked about before. 543 patients with suspected Lyme, no sign of this acute relapses of relapsing fever. 32% had Lyme, 22% did not have Lyme, they had relapsing fever, and 7% had both. Okay. Like I said, the possibility of a negative Lyme test may be because you're testing for the wrong thing. None of the commercial test kits, from the big box labs or from the hospitals that test for Lyme, none of them, whether the IFAs, ELISAs, Western blots, PCRs, even the T-cell tests, have been validated for all the different Lyme Borrelia, that Borrelia burgdorferi, Sensolato, that whole list I showed you, or for any of the relapsing fevers. Okay, if you test for relapsing fever, they only can test you for 2 species, that Hermsii and Miyamoto, and each test has to be ordered separately. There aren't commercial tests for the other ones until IGeneX came along. So they made the immunoblot not only for Lyme, but for the relapsing fever. So they found that you can detect the most commonly found ones, not just these 2 Hermsii and Miyamoto ones. So that means it's much more sensitive. You're picking up germs that other tests can't pick up because they're not designed for it. It's a better test. It's more sensitive. It's more specific. And as I said before, there's no cross-reactivity. So if you have a negative Lyme test or a positive relapsing fever test, then you're pretty sure it's relapsing fever and not Lyme. How about validation here? Well, again, early case, they found 2/3 were positive, which was supposed to be 0, but in the immunoblot sense, they found 2/3 positive. In late case, 100% were positive. And these are 15 patients, all of whom had positive PCRs. They also found that the IgM was positive in a number of these patients, and going to the specificity, 98% specific, including IgM. So once again, a positive IgM in relapsing fever, just like in Lyme, is not something to be ignored, even in late disease. So if you're considering Lyme disease, you really have to consider using tests that have the highest sensitivity, highest specificity. They're not going to give you false positives, and include broad species coverage to get all these other germs that are out there that no one had thought about before. So that includes the immunoblot and the culture, not the IFA, not the Western blot, not the ELISA, and the 2-tier testing and all these other protocols that are meant for surveillance. Don't waste your time. They're not for diagnosis, okay? Now, as I showed you, sometimes people's immune systems are compromised, and the serology test, even a good one like an immunoblot, may not show it. And that's why people are now doing the culture at the same time that they do the immunoblot, because you're directly testing for it with the culture, and you're looking indirectly through the immunoblot. Another option you can do, if it's involving your joints, you can actually biopsy the joint and culture that too. So there's a lot of new options here. All right, we're going to move on to Bartonella. Bartonella is a big problem in Lyme patients, as a lot of you know. First of all, it's been documented in at least 49 states. And again, the map is showing positive test results that IGeneX got. This is not some survey of animals or looking at insurance claims. This is the actual cases that were positive at IGeneX. So they found it in many, many Lyme patients. It's very easily confused with Lyme. Now, just like with Lyme, it's not just one thing. It used to be called cat scratch disease, and that's one specific version of Bartonella. But they now know there are at least 45 different species. And if you don't have a test for all 45, you're going to miss it. And why is it so common? Well, it's easy to get. Fleas, mosquitoes, biting flies like deer flies and horse flies, mites and red ants all have been known to carry and transmit Bartonella. I know a couple of years ago, I got a really bad case of Bartonella from being bitten by the deer flies. They also now have shown that ticks can carry and transmit Bartonella. It was controversial to say, well, you can't get Bartonella from a tick bite. But then they said, well, we found it in ticks, but you can't get it from the tick. well, that's silly. If it's in the tick and the tick injects its contents into you, of course you can get it. And sure enough, they found you can get it. Also, you can get it from animal bites and scratches. That's why it was called cat scratch disease. And in fact, two recent cases of mine both got it from cat scratches and one from a dog bite. Needless to say, veterinarians and animal workers have gotten Bartonella from that. And Bartonella, like Lyme, can be transmitted from mother to baby. And here, look above the Arctic Circle. These are all carried by birds. Look at Hawaii even. So it's all out there. Now, how do you differentiate Bartonella from Lyme clinically? Well, aside from the general symptoms that's shared with Lyme in terms of being fatigued and headache and feeling poorly, there are specific things that make you think of Bartonella. One thing is that it's a very strong irritant to the central nervous system. So people can have the brain onset that they've never had before of anxiety, panic attacks, even to the point of antisocial behavior and rage attacks, can be insomnia that you never had before, can cause mood changes, depression, can cause tremors because it stimulates you, can cause seizures and even what are called pseudo seizures that look like seizures but don't show up on testing, can give you an unsteady gait where you feel like you're like dizzy all the time, can affect your mood with depression, can give you hallucinations, even mimic schizophrenia. It can involve the eyes, can inflame every layer of the eye, and including blocking the arteries and veins. This is a common thing that eye doctors see. Unlike Lyme disease, it can cause swelling of the lymph glands. And I had a patient a number of years ago who was about to start chemotherapy for lymphoma. Turned out it was Bartonella, treated with Bartonella, the lymph glands shrank away, and it wasn't lymphoma. It's a false reading from the lymphoma testing. The thing about this, though, is that when it gets into tissues, it gets into what are called the connective tissues. In other words, it doesn't hit the joints with the thickening around the joint, around the knee, and around the nodules, around the fingers. It actually gets along the ligaments and tendons. It can go along the sides of your legs, on the back of your arms, where it's all sore, tender nodules. On the bottom of your feet feel sore in the morning when you get out of bed and trying to walk. And again, it's more in the tendon, not in the joint. You can feel like pain in the bones. And the joints can be painful without being swollen. And that's the difference from Lyme disease. Bartonella is also known for strange things on the skin. They look like stretch marks. Then they said, well, that's not accurate. We'll call them Bartonella tracts. And now they call them BACL, Bartonella associated cutaneous lesions. I'll show you pictures. They also can form what look like pimples that can scab. Another thing Bartonella makes it different from Lyme is that it can cause stomach upset, just like ulcers. It can actually mimic H. pylori. It can cause belly pain because it infects the lymph glands of your intestinal tract and can also infect the liver. So here are pictures of these rashes. Look on the follow the arrow. The thing about this is, unlike stretch marks, these are red, and unlike stretch marks, they don't follow skin planes. See, this is not a stretch mark. These are not stretch marks. See, this looks like somebody leaned against, a wooden fence or a lawn chair. And here this person has both the tracks as well as the red bumps. These are some better pictures of the red bumps. See, so that's bacillary angiomatosis, and these are the Bartonella tracts with the BACL. Testing. Big box labs, the commercial labs, the hospital labs, they do what's called an IFA, and it's only for cat scratch disease, which is one species, Bartonella hensleyi. You don't get testing for the other ones unless you specifically ask for it. Then there's only maybe one or two others that you might, if you're lucky, be able to get. So IGeneX has an immunoblot for that. And just like with the Lyme, it's more sensitive, it's more specific, and can detect a whole bunch of different species, not just that one. You can get a PCR for Bartonella. A lot of labs offer this. Their own studies that have been published show it picks up only 6% of the cases. In other words, 94% of the time you get a false negative test. That's why the PCRs are so poor. The culture, though, for Bartonella, just like with Lyme, has been found to be at least 10 times more sensitive. And in fact, as I looked at the data, it's probably much more than that. Also because you're looking at the genus level of Bartonella, what that means, it's not Bartonella hensleyi or Quintana, it's all Bartonella. It allows you to pick up pretty much most of the important infections that people in America have gotten. So the culture is a huge advance, and I'll show you pictures or studies on how the culture has changed things. Another test, which is really neat, you can get from IGeneX, is called the FISH test. It stands for fluorescent in situ hybridization. It has nothing to do with going out with a fishing pole. But what they do is they take a drop of your blood, they put it on a blood smear, look under the microscope. But instead of just using a stain or a dye, they use a probe that is fluorescent and attaches to the RNA, the nucleic acid of the Bartonella germ. So it allows them to see with the microscope, with their own eyes, there's the germ, and it lights up like with the black light. It's like very, very obvious. And studies have shown this to be at least 100 times more sensitive than a standard blood smear for looking at Bartonella and even in some cases Babesia. I'll show you that later. Again, the fish looks at the group of Bartonella as a genus, as a whole inclusive group, not just individual species. And what's really important is it can detect even if the organisms are hidden in biofilms. Biofilms are a big problem with Bartonella and also Babesia because they can exist in the bloodstream and make the blood test not show these up. But because these tests will go through the biofilm, you can actually find it. And this has been shown way back into the 1990s, was first discovered that this can do that. And that's why the fish test has become so very popular among the doctors. Now, the other thing that's really important, RNA is made fresh by a living germ. As soon as the germ is dead, the RNA disappears within minutes. So if you have a positive fish test, which is a probe for the RNA, a positive test means the day the blood was taken, you had that infection. So it definitely shows an active current infection. Now they did an immune blot validation study, and here they looked at people who had Lyme and relapsing fever, Lyme and Babesia, Lyme and Ehrlichia, Anaplasma, Babesia, and negative controls. And in none of these cases did you get a false positive Bartonella immunoblot. So the specificity in this series was 100%. Here, when they found people who had a positive Bartonella, but either fish, IFA, or Western blot, any one combination, every single time it picked it up. Some were IgM, some were IgG, but if you looked at either one, and you can, because as I said, they're both 98 to 99% specific in the other testing here 100% of the time, you can combine them and they picked up basically every single one. So this one validation study is a good idea that this is really a good test. The fish, they tried to see what would happen if you tested a fish with other infections. Didn't cross-react with Babesia, either Microtia duncana, didn't cross-react with Lyme, with malaria, with trypanosomes, Anaplasma, Ehrlichia. All these other ones were tested. Not a single time did you get a cross-reactivity. So it was very specific, didn't get a false positive reading. On the other hand, they tried different Bartonellas, Vincenti, Hensley, even some unidentified species, and it detected all of them, including the one that was unidentified. They picked it all up. So it really showed a good performance on the fish test. So the recommendation is those who know about Bartonella know it's a very hard thing to pick up in the laboratory because none of the tests are 100% sensitive because they are stealth infections. So between that and the fact that there are many, many species that are potentially infecting people, you need testing that includes very high sensitivity, high specificity, and coverage of a lot of different species. So just like with the others, you have to combine testing. We like the immunoblot because it's the best indirect test. Fish and culture are the best direct tests, and they are very broad in their coverage. Well, sometimes people are immune deficient, and you can tell they have a B cell that doesn't work so well because their B cell levels, IgG levels are low. You can add the T cell response, and James calls that an IGeneX spot. You can add that to the immunoblot to get a better picture of what's really going on. So there are many options now for Bartonella. Babesia. It's a parasite. It's not a bacterium. It lives in the red blood cells mainly, although it does swim freely sometimes. As I showed you before, it's the most common co-infection in Lyme patients. And what makes it different from Lyme is that it can cause fevers that are significant, real sweats, not just a little clammy, but really, really sweats. The headaches in Babesia, unlike Lyme, which is usually the back of the neck where you feel like you have to crack your neck all the time, this is more like a migraine, the pounding, hurt the eyes with light kind of headache. And interestingly, even if it's Babesia migraine, it still responds to migraine medicine. So just because you have a migraine medicine and it goes away from that, it doesn't mean it's not Babesia because it mimics that. The other strange thing it does can cause air hunger. Like you just do nothing, you feel short of breath. You're talking on the phone and you're losing air. It's like to take a breath all the time. That's called air hunger. Babesia does that because it congests the flow of blood through the lungs. The same reason it can cause a cough for no apparent reason, a dry cough. Because it's a parasite, you can get really profound fatigue. And because it blocks blood vessels, just like in the lungs, it does that in the brain. It can cause strange sense of being off balance. Now, it's not vertigo, you're not spinning, and it's not lightheaded like when someone has POTS. It's different. It feels like you're going to tip over. People go on boats for a long boat trip and they get off the boat on dry land, they still feel like they're swaying. It's kind of like that. It's like you're walking up the stairs and feel like you're going to fall over backwards and fall down. It's this very strange feeling of imbalance. Those are the ones that are distinguishing Babesia from Lyme, but many of the other symptoms, like the fatigue and malaise, that overlaps with Lyme and relapsing fever. The thing about Babesia, because it's a parasite and because it also hurts the immune system, if you have Lyme at the same time, it makes the Lyme much more severe and much more treatment resistant. So if someone has Lyme and they're just not responding well and they're really sick and you're treating a Lyme and they're just really not doing well, even if the tests are negative, you have to consider Babesia and then maybe order the cultures in fish and immunoblood to really be thorough, because that's a clue. If the Lyme is really, really tough, you have to think Babesia. And the thing is, it can be transmitted by the same tick that transmitted Lyme, so it's common. Now, in America, the two main ones are microti and duncani. They used to call duncani WA1 because it was first found in Washington State. But there's also MO1, which is found in Missouri. There's otocolii, which is the most common Babesia found in deer. And people are thinking it's probably in humans as well, and that definitely has been found. The rare diversions is thought to be in animals and not humans, but that's also seen now, especially with the culture. And even atypical species, I'll show you some important example of that. They're finding things that you never thought were found in people. So how do you test for Babesia? Well, if you end up really sick and end up in the hospital with an acute case, they can do a blood smear and take a look at with a regular stain, and they see it. But it's only useful in the first week when there are a lot of germs in the blood before they get cleared out by your body's immune system. So that's only something done acutely and in hospitals. It's really not useful after the first week of infection. You can do an IFA. Again, the big box labs and the hospital labs do this. IFAs are insensitive, they're outdated. You have to have a separate IFA for microti, a separate one for duncani. You can't get it for the other species, so then you're at a loss. But you can get a fish test from IGeneX. Again, it detects the RNA, so positive means that it was living the day that you had the blood done. And studies have shown that it's at least 100 times, if not 1,000 times more sensitive than a standard blood smear. As I said before, the fish can detect them even if they're in biofilms. Because it's genus level, it can detect all different Babesia, not just the microti. You can get immunoblood now for Babesia, and it's a favorite for a lot of different doctors. It's far more sensitive than the other testing that's been out there. And again, multiple species. Sounds like a recording, because this is the whole point. You need to look for everything. And the culture, you can now get a culture for Babesia. Not going to detect microti and duncani. It's picked up some really strange things. Let's go through the validation. Because you're using multiple immunoblot antigens from multiple species, you get a lot of sensitivity and specificity, and you can detect a lot of different species. Here's a test for sensitivity. They found the overall sensitivity was about 95%. They picked up all the duncanis, 89% of the microtis, and together you combined for 94.9, 95%. In terms of being specific, they said, well, how does it turn up if you have anaplasma? Will you get a false positive immunoblot? No. Now for Ehrlichia, anaplasma, Rickettsia, Lyme, and normal controls, 15 of them, 100% of the time it was very specific. So a positive immunoblot means you have it, or at least you've been exposed. You can get the culture. As I said, they're picking up some really strange things, and this is a case that kind of is pretty historic. It's a case report of a woman. This just happened last year, 46 years old, 4-year history of diagnosis of chronic fatigue syndrome. Nobody knew what was wrong with her, so they labeled her chronic fatigue syndrome. They test for Lyme, relapsing fever, but now all the others were done, nothing showed up. So they did Babesia culture, and they found a thing called Colpodola. They said, what the heck is that? They didn't believe it, so they had the test run a second time, and it was Colpodola. This was sent out to an outside lab, not once but twice, for sequencing. Colpodola is a free-swimming organism. It lives in ponds, sometimes found in the soil. In the whole worldwide literature, there's only 2 other cases of human infection of this Colpodola. And guess what? Another one was found, and how was it found? With the culture from IGeneX. Luckily, No one knew what to do with this, but because Copperdoll is in the same family as malaria and babesia, they gave the patient Metron and she got better. So the point is, there are a lot of different things out there that we don't even know about. And now that we have these better tests, we're finding it, and now we're getting people better. Now, I just want to show you how sensitive and useful the culture can be. They call the culture CEPCR, culture enhanced PCR. Here's a patient, again, immune deficient, positive culture, negative immunoblot. Just like with the Lyme and relapsing fever, you can have someone whose immune system is either impaired or damaged, and they can have a positive culture even with a negative immunoblot. Here's one, positive Duncani, negative immunoblot. A patient was immune deficient. No diagnosis would have been made if that person did not get the culture. So the recommendation is, because again, it's notoriously difficult to detect, there's no one test that's 100%. And now we're also finding really strange species that are not expected to be found in humans, and they're finding them. So you really want to get testing that is very sensitive, very specific, very accurate. And that's why we say using complementary methods, an indirect test and a direct test, a FISH, a culture, immunoblot. These are the three that you do. And just like with Bartonella, if there is a B-cell immune deficiency, you can add the IGeneX spot, the T-cell test, and that might pick it up when the B-cell test does not. We're trying to outdo Mother Nature here, and it's tough, but you can do it. We're going to finish now by talking about the rickettsias. We're actually seeing more and more rickettsias in the patients, and it's not surprising because anytime you look at articles where people collect ticks from the wild and they look at what's in them, some of them have Lyme, some have relapsing fever, some have babesia, but they seem to all have some kind of rickettsia. That includes Anaplasma, Ehrlichia, and the rickettsias are all part of that same rickettsia family, as is Rocky Mountain spotted fever. Clinically, this also causes an acute onset. Headache here is not migraine-like it is with babesia. It's more like a knife in your head. It's really a sharp, terrible pain. And here, it's not the joints or the tendons that's sore, it's the muscles. That's what myalgias mean. And again, the really sick malaise feeling. Here are clues that you can have low white cell counts, low platelet counts, and sometimes liver enzymes are elevated. So this is a clue a lot of doctors will do. Someone comes in really sick with a high fever, you expect a high white cell count and sometimes high platelets as a reaction to the infection. But if it's the opposite, if it's low, then you can start to think about this. Now, Rocky Mountain spotted fever is called that because it gives you a rash, not 100% of the time, but at least 80, 90% of the time. And the rash has two unique features. One is that it can be on the palms and soles. And usually rashes don't do that, but this one does. The second thing is it consists of red spots, as you can see, and the spots have like a little red center and gets kind of faded as it goes out to the edge. If you were to put your finger and put pressure on that, the spot would blanch. And as you take your finger off, it would come back pink again, but it would refill from that center dot. So it's like it refills from the center vein, because this is what's called a vasculitis. It inflames the arteries and veins. And that's a characteristic way of diagnosing it, by just putting your finger on the rash. Now, this kind of rash you can find in Anaplasma and Ehrlichia as well, but not that commonly, less than 5%, but it does happen sometimes. Testing for this is tough. The only serology available at this point is an IFA. It's old-fashioned technology. They don't have an immunoblot yet for this. But they do have a culture for Ehrlichia, Anaplasma, and the basic ricketsias. And used to be a PCR, but now they got rid of that. You have to do the culture. It's much, 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 much better. Unfortunately, for Rocky Mountain spotted fever, the government doesn't allow culturing because it's a biosafety level 4 type of a germ. So you're only stuck with the standard PCR and the IFA. But the best advice is to use all methods you can, because these can be very, very acute, and people actually die from this acutely. So it's really important to do every test you can, because you don't want to miss a case. Here again, positive rickettsia culture, negative IFA. The IFA is a really bad test, but if it's all you got to do it. But here we have a culture that's short. Now look at the result. What was it? Rickettsia rickettsii. That's Rocky Mountain spotted fever. That's something you're not supposed to be able to pick up on a culture, but they actually found it. It was missed by the IFA, but it was picked up by this one. This anaplasma was positive, despite a negative anaplasma IFA. So the culture showed human granulocytic anaplasma. Okay. Here's one that showed Rickettsia felis. Rickettsia felis, that's an infection that's found in cats, not supposed to infect people. But they found, in fact, was positive on the culture, proven by the special PCR. So here we found once again, not only was it positive when the serology was negative, it's actually a species no one ever expected to find in a human. All right, so the conclusion, I only ran out of time a little bit. It's vital to use test methods that are inclusive of all the different species you know that you can possibly test for, because just a few tests are available from the big box labs. It's a waste of time. Don't rely on outdated and restrictive test methods. Don't use surveillance criteria when you have a diagnosis to be made. And realize that the tests from IGeneX, they're more sensitive, more specific, inclusive of more species. And the highest yield is when you combine the different methods that are complementary. Now, I apologize for this being so intense, but like I said, I wanted to show you this proof that these beta tests are important, that they work, that there's studies to validate that they are accurate. And some says, oh, IGeneX test positive everybody. Well, no, it's been shown to be 100% specific or close to that in every single case along here. And they're showing things that no one ever expected to find, including animal germs and pond scum infecting our patients. All right, so I think at this point, we're done. Hey, so I'm going to give the screen back and let you guys bring up some questions. How do I make that? I guess I don't do that.
Click on the share screen. I think you can turn it off.
Which one do I check on? I'll stop share.
Yeah, stop, correct.
Ah, there we go.
Yeah, there we go.
All right, all right. Okay, so are you still okay? Did I put you to sleep?
So if any of you have any questions, you can go ahead and ask them out loud, or if you'd rather write in the chat, one of us can read it. But yeah, thank you, Dr. Burrascano. That was really interesting.
Oh, you're welcome. Like I said, I really packed this full of hard information, and I think it's important to get a copy of the slides. So if anyone starts to doubt you or what your Lyme doctor is saying, and you know there are a lot of doubters up in your neck of the woods, you have the data to back it up.
Yes, I just got my results back.
Oh, I'm sorry.
Oh, I just got my results back from you guys, and everything came back negative. But I'm still really, really tired.
Well, did you have the different types of testing done for the different types of germs? Were there any that you missed?
Excuse me?
Are there any types of the germs that I talked about today that you did not test for? Did you do the testing for all the different kinds of germs that are out there?
I just sent in for the blood test for Lyme's. My fish came back negative. You know, everything came back negative.
Well, two things. One thing is, if you like, you can share your results with Johan, and he and I can go over it. We'll get back to you and see if there's something that is suspicious or a test that should be done that wasn't. So that could be one thing. The other thing is that, you know, these infections, they really take a toll on a person, and they do a lot of other things besides just cause an infection. They damage your metabolism, they impact your hormones, they prevent your body from removing standard toxins, and you can build up mold toxins and heavy metals and other things. Because they weaken the immune system, you can have old germs come back and reactivate. A lot of times chronic fatigue is a result of a tick infection, but it becomes a reactivation of mono, of Epstein-Barr. And so part of the evaluation looks for these kinds of things being reactivated. So there's always a reason for things. And, you know, a good Lyme doctor will go through the whole list and help you figure it out. But in the meantime, if you like, you can have Johan, you know, give him a copy of your test results. And if you want to take your name off, that's fine. And we can figure out if there's anything we can do to help you out in that regard.
Okay, thank you.
You're welcome.
Yeah, I'll share my email too, and I'll have it. You guys can email me questions as well, or if you have things like that you want to go over with me, I'm here as a resource. So
We had
I have
one
a question.
Okay, go ahead.
Dr. Burrascano, first of all, thank you for all you taught me. It's just really, I brought it back to Minnesota and spread it around, and you know, it changed my life and changed the life of all the people here. So heartfelt thank you for that. And
You're welcome.
I can't believe you presented a whole complete talk on Lyme and all the co-infections in such a short amount of time. So that was amazing.
Yeah, but that's why everybody's dizzy right now.
No, but you present it in a way that people can understand it, and nobody else can really do it like you can.
Well, thank you.
But-
Thank you very much.
Anyways, when there was a culture test previously that you were associated with, when we sent cultures in warm weather, we'd get positive results, but when we sent them in Minnesota winters, we'd get negatives across the board. So is that going to be a problem with IGeneX cultures, with the shipping in the cold weather?
What was done was they tested not only the accuracy of the culture test, they tested the shipping. And what they did was they took samples and they sent it to themselves. And they tried different things, including like those ice packs and other things. And they found that they had to put special packs in different weather conditions that weren't so cold that they froze it or so hot that the thing got unstable. But basically, they did work that out. And as far as I know, it seems to be okay. So if you find that there is a seasonal discrepancy, definitely let us know right away. That, Johan, I don't work for the company, so don't tell me, but I could tell them,
No.
tell Johan and we'll figure it out.
And I haven't heard any of that at this point, at least. So
If there's more negative blood cultures in the winter than in the summer, you haven't heard anything like that?
no, I've not heard. I mean, the testing has been out now 18 months or so. So I guess we've gone through at least winter and a half, if we call the last one a really hard winter. So we might need some more time for that. But so far, that's not anything I've heard being reported to us that there's been a difference. So, or providers that have complained either. So
And is there a panel for all of these
yes.
tick-borne infections that are common in Minnesota?
Yeah, we do for the culture test. We have there's 3 different panels. You can order them all individually, but the cost of the culturing itself is $500. So if you're gonna do them separately, it's better than to do them at one point, and then you get a much better price break by ordering one of our panels. So I think the most expensive one we have, including all the different pathogens we look for, is like $1,800 or something like that.
So that's pretty
So-
good. Thank you.
Thank you. And it's all available on our website. We have all that information on our test requisition form. That is, anyone can go and get off our website.
Well, and that kind of ties into one of the questions that's in the chat. One person said that it's hard to fill out the IGeneX test order forms with so many options. Will there ever be a way to add a symptom checker feature so that people could put in what they're struggling with and it would recommend testing? I'm waiting for one quote unquote kitchen sink test that will screen for all tick-borne diseases. It's so expensive to order more than a couple.
Yes, no, I mean, absolutely. One of the, that's one of the feedbacks we get all the time is that our testing is expensive, and we know that, and there is a reason for that too. And I think Dr. Burrascano went through a lot of the research and things that go into developing our test, and that's part of why it's very expensive. It's a little intimidating with our test rec, but we have, backside is all our individual tests, and then we do have some of the panels. And the reason there's so many specific doctors like to have their panel on for their patients. And I think most people there's a few of them that most people are using. I mean, one of the more comprehensive one is our TBD6. I think we call it TBD6 IBL. And a lot of people that are on Medicare and would be eligible for that use that one, because that's the most comprehensive one we have, which has a nice balance between all our immunoblots, fish testing, as well as some other indirect testing, the PCRs. We don't at this point have a combination between the culture enhanced one, but maybe that will come in the future. We'll do one big one, but cost is also really prohibitive on that side. It is good to know that we do save the blood for 60 days. So many of these tests you can then add on afterwards, and you can do one at a time to spread the cost out somewhat. So, and I always encourage people to fill out third-party out-of-network the insurance coverage. So we will then electronically file that for the patient, and people do get money back. We don't see what comes back. That's why we don't have a good judgment of exactly what people are getting back. But if they have fairly decent insurance, they will get, in many cases, at least I think last I heard is 75% will get some money back or get a payment towards their deductible. So.
And does Medicare cover all IGeneX tests or just certain
So
ones?
Medicare basically, as long as you don't have a, well, the HMO-based Advantage plans are the ones that are not covered all the time. And I think overall, the patients that we see, that only is about 15%. Most other straight Medicare, et cetera, should be covered. And the only thing with our big panel, the TBD6, is that there is a co-payment for the fish testing at $44. So that whole big panel would be $88 copay. And that's billed after the fact. So, and there's no one that for Medicare that we do the verification when the testing comes in. And if someone doesn't have the right coverage, we will let them know. And so they're never have to be charged any money for the Medicare side of it. So just to make it, we can't do that by law. So I hope that helps them.
There's quite a few questions. The next one that kind of ties in, can a physician order IGeneX tests even if they're affiliated with a hospital? This person thinks there might be some confusion on this.
I mean, anyone can order it from us. We have no restrictions on what doctor can order it from us, as long as they have an MPI number and are medical certified. But certain medical systems don't let their doctors order testing if it's not one of the ones that they have pre-approved. So, but that's a hospital issue, not an issue by IGeneX at all.
I think what can be done also is that there are a number of different blood drawing services that the doctor can just, sign up with them separate from the hospital, and they'll meet the patient, even in their house and do the blood there. So there are ways around it. If the doctor is really interested, they can contact Johan and figure it out.
Yep, absolutely.
Britt Gina had a question that might have scrolled off.
Up towards the top. Do you have
Yeah.
many people that do you have many people that get tender nodules on the breast with Bartonella? Is there a way to test the tissue if you have a mastectomy?
Oh, absolutely. In fact, it's been known that Bartonella can infect people with breast cancer and make the breast cancer much worse. Because Bartonella infects the blood vessels and the other lymphatics that drain where the tumors go. So definitely there's an association. I don't know if it's like a chicken and egg situation, but definitely they are somehow related. And you definitely can do a test on a biopsy of the breast or breast tissue, and that would be the culture test.
Thank you.
You're welcome.
Yeah, and if I'll add to that too, if you're going to send tissue in, make sure you reach out to us in the lab so we know it's coming in, because it gets handled a little bit differently when it gets into the lab, so we know it's coming. So I'm available for all those consultation. I'll usually put you in contact with Iris, who's at our lab and handles that. So
Thanks.
you're welcome.
What kind of test is it that they use for Babesia when you give blood?
I don't know what they use. I'm sure it's not a good test. I think they use an IFA, and I think they use a smear, but I'm not completely sure, because the smear requires someone to sit and look at the blood under a microscope for anywhere from 10 to 20 minutes for every single sample. So I think they run an automated test like an IFA. And as I showed you, it's a very bad test. You can miss a lot of cases.
So
Yeah,
I
it's-
hadn't even thought of this question until I just asked this one, but I've been sick now for 20 years, and a number of years ago, I took myself off the donor list and I no longer donate blood just for the rest of my life. And I feel like I don't want anyone else to go through what I've gone through, but is that recommended, or is that? I mean,
Yeah.
I've had people say like, well, if your testing is so expensive, just go give blood and then they'll tell you what you have, which I don't, like you're saying, I don't think that's a good way, but
No.
Yeah,
No, they only test for Babesia. They don't test for Lyme. They don't test for Bartonella. They don't test for Anaplasma ehrlichia. They don't test for any of those things. And in fact, there's a condition, it's called TRALI, T-R-A-L-I, transfusion-related fever, illness, or something like that. And it's because the blood cells were infected with a germ like Bartonella or Anaplasma that the person got. And after transfusion, the guy who got the transfusion suddenly gets sick. And they recognize it's from an infection transmitted by the white cells. But they don't go one step back and test the blood for these germs. But think about it, there are thousands of germs, you know, they'd never be able to do it.
Scary.
Yeah, don't get sick.
Brett, I'll ask, I have a question about Bartonella. Recently we did some ads with just for some awareness ads, and I was looking on the Minnesota Department of Health site for tick-borne diseases, and Bartonella wasn't mentioned on the list as a tick-borne disease.
You know, that's why I mentioned that specifically, because a lot of people still claim it's not tick-borne. Now, truthfully, it's probably not the main way people get Bartonella, but it's still a possibility. Also, you know, where there are ticks are also the kind of bugs that bite, like fleas and biting flies, and they're much more likely to transmit it because you know how biting flies are. They keep biting and biting and biting until you finally smash them away. Unlike a tick, which, you know, has to crawl on you and find a spot and so forth. So it's much more likely to get it from a flea, from an animal, or biting flies. And fleas do live in the grass too. So, you know, that's more likely. But it doesn't mean you can't get it from a tick bite. And now there have been articles that prove you can.
And some of our sickest patients have had Bartonella, but established medicine thinks that Bartonella is a simple disease that comes and goes by itself without being treated, and no treatment is needed.
Here's another question. Is the treatment for Lyme disease different than that for relapsing fever?
Well, generally not, but there's one difference. As far as we know, the same antibiotic regimens for one will work for the other. But some people have a more acute form of relapsing fever than they would have if it were Lyme itself. And the Herxheimer reactions can be much more severe. So if someone is known to have relapsing fever, the recommendation is to start treatment more gently because it could be a stronger Herxheimer.
Okay. When can a positive serology be considered a positive diagnosis? I know of a specific instance where the person had a severe chronic illness. Bartonella is strongly suspected, but the only positive on an IGeneX panel 51 was IgG for B. myomodi. The positive serology has not been considered as a positive diagnosis.
Well, you know, as I showed you, a positive serology is at least 98% specific, so that there's less than a 2% chance that that's a false positive. Now, the question is it a current infection or a past one? You know, a serology test positive reflects exposure, not current infection necessarily. So it all goes down to the symptoms. And again, you need a good Lyme literate physician who recognizes all of this. Practitioner, it doesn't have to be a physician, could be a PA, nurse practitioner, but someone who's Lyme literate, who knows how to figure it out. But things happen for a reason. If you have one test that's positive of a germ that's known to make you sick and chronically sick, I mean, you know, why ignore that?
If you tested positive several,
Oh.
I'm getting some feedback.
Yep.
If you tested positive several years ago, can you test now to see if you still have an active infection, or does the test show that you have had an active infection at some point?
That kind of depends on the test. If you're doing a direct test like a culture or a fish, or even a PCR, those look for the germ itself. And if that's positive, then you have it. But those tests only work if the germ happens to be in the blood the time you took the test. And it's like I said, it's like going fishing. you don't always catch a fish, but it doesn't mean there's no fish in the water. You just have to pick it up. On the other hand, a serology, like an immunoblot, that stays positive while you're infected. And if the infection's cleared over a period of months to years, it does go away. So if someone has been off treatment for a long time, a matter of years, and test still shows positive, there's probably, at least in my mind, still some lingering infection somewhere. And it is possible to have a subclinical infection where you basically are living with the symptoms and your body's kind of in a balance where you're at a stalemate with the germ and you're pretty much okay. The only danger to that is that it is possibly transmissible to others through pregnancy, blood transfusion, and possibly through intimate contact. So that's the only danger of that.
Okay, and I think we have one last. Oh, here. My husband had a positive immunoblot for Lyme, but was inconclusive whether it was current. Do you recommend a fish or culture test?
Well, for Lyme, they don't offer a fish test because that's not something that shows up on a blood smear generally. The fish is for Babesia and for Bartonella. So for Lyme, to look for specifically if it's still present, actually you have two options: the culture test, which is the newest one, and the other test, which is the oldest one, happens to be the urine antigen capture. I don't know if you know about that, but IGeneX started offering that test probably in 1991 or 93, where you collect urine on 3 different days and send it in, and it can pick up the Lyme. Turns out that people and animals will spill Lyme germs and fragments of Lyme germs into the urine, especially on days when they have a lot of symptoms. And so if you are having symptoms that come and go, or if you start medicine and have a Herxham reaction, or if you have your menstrual cycle and be worse when you're cycling, those are the days you can collect the urine and send it off. And generally, we collect 3 separate urine samples, let's say day 1, 3, and 5. And any one that's positive, you believe, and a negative, you don't necessarily believe. So that's another way of doing it. And it's also a really good test for newborns. If the mother has had Lyme disease and now has a baby, you know, you want to test the baby, but you don't do an immune test on the baby because the immune system hasn't matured. And you don't want to take a blood test on the baby on a regular basis because that's hard. So what I always would do would be collect a urine test on the baby at least once a month for the first 6 to 12 months. So that's another option too.
Good. And then I think the last question we have so far is: a family member of mine had an acute illness last May that was treated with doxycycline and then developed severe retina problems resulting in vision loss. Which test would you recommend to see what the root illness might have been?
Well, of the tick-borne tests that I mentioned today, the number 1 for that would be Bartonella. And because it's a very important diagnosis to make, I would do everything: the immunoblot, the culture, and the fish. In some people, Babesia can do that too, because Babesia can cause blockage of the small capillaries. And that's why you get air hunger and the dizzy feeling and the spacey head. And it's possible that can also happen in the eye. So definitely Bartonella testing. And I probably would add to that Babesia as well. And again, the immunoblot, the fish, and the culture.
Oh, here's another one. If Bartonella is strongly suspected but tested negative on all IGeneX tests except CE-PCR and X-spot, which weren't performed, the person is on steroids. Which test would you recommend? The person has the tracks and all the Bartonella symptoms.
Well, the culture would be the obvious one, the culture and the fish test, because both of those are not affected by steroids. In fact, they might be even more likely to be positive. But, you know, all of these things are clinical diagnoses, and having the tracks, that's the diagnosis right there.
Great.
I think we've done it.
All right. Great.
And we all survived the storms. I don't see anybody running from the basement yet, so that's good.
It's quieted down where I am.
Wow. Yeah. Yeah, one of your storms came to us last week. We had 5 inches of rain in a matter of, a few hours. And now, that was last week or the week before. Now my grass and my trees are growing like crazy. Oh my God.
Yeah.
I just hope the ticks aren't growing, that's all.
Same up here. So yeah, and if anyone, Britt, I'll share my contact information with you, and you can post it to the rest of them if they have any questions, follow up, etc.
Okay.
For me. So and thank you for having us. I think this was great, and thank you, Dr. B. You're always great on these talks and put the level where we need to be. So thank you, everyone.
I appreciate it. Thank you all for your time.
Yeah,
Thanks.
Yeah. We
Thank
really
you.
appreciate it.
All right, we'll sign off. Thank you all.
Thank
Good
you.
night.
Thank you so much.
Have a good night, everyone. Thank you so much for your time, Johan and Dr. Burrascano.
Thank you.
Thank you.
Bye-bye.
Thank you.
Bye.
Thank you, everybody.